to see about the immunity. Immunity is defined as the resistance of the body against any pathogenic agents. So if any pathogenic agents or any agents are coming inside the body will respond to that particular organism or that particular substance, and that will help to remove that substance from our body. That harmful substance are removed from the body. So the immunity is classified into innate immunity and acquired immunity. Innate immunity means immunity that is non-specific defense or natural killer cells defense. There are of two types. They are non-specific and specific. So by the word meaning itself, innate means by birth itself we are getting that particular immunity, like our saliva or our skin or some like that. The immune system is known as innate immunity. And acquired immunity is actively acquired and passively acquired. So again, immunity is innate immunity, acquired immunity. The inacquired immunity, again, further classification is there, like passive immunity, active immunity. Active immunity is again further classified into humoral and cellular immunity. So natural or innate or non-specific immunity means it is present since birth, which prevent the entry of microorganisms into the body. So maybe specific to a organism or non-specific to a particular organism, and which prevent the entry or which destroys that particular substance. Like phagocytic activity of WBCs, because WBCs are present in our body by birth. And neutrophil macrophages, natural killer cells and all. Acid in stomach and enzymes in the gastrointestinal tract. That acid, the acidic content will destroy the microorganism which it enters through the food. Lysozymes present in the tear and saliva. Also the protective functions of skin are known as the natural immunity. Acquired or specific immunity, like resistance developed against any specific foreign body. They are most powerful immune mechanism. They are like active immunity by humoral cells or cell mediated, and also by passive immunity. And the natural active immunity, that is the immunization, is acquired by activating the immune system. They are through infection, or artificially we can achieve that by taking vaccines or toxoids from the bacteria. Passive immunity, that is the administration of serum or GABA globulin from a person who is already immunized. That means passive natural, like the maternal antibodies are protecting us. Also the passive artificial, that is injecting antibodies from other sources, that is anti-tetanus serum or anti-diphtheria serum, like that. So immunity is active and passive. So going to the immunity, we have to know what you mean by antigens. The term antigens are the substances that stimulate an immune response in the body. So, which has some characters like immunogenicity, antigen specificity, and species specificity, also isospecificity. The isospecificity is just like blood group antigens. So this will stimulate a particular immune response. If it is enters into the body, it produces some type of immune response in the body. And MHC, that means the major histocompatibility complex. Histocompatibility complex are the genes which encoded by the genes of antigens which are present in the plasma membrane of cells of each individual of a species. They are a complex genes, and they are known as monohistocompatibility complex, or MHC, which is located in chromosome 6, and MHC in the surface of leukocytes are known as HLA, that is human leukocyte antigen. So MHCs are classified into three, that is MHC class 1, which is present in all type of cells except RBCs. MHC class 2 are present in immunologically reactive cells, and MHC class 3, which is present in genes coding for the classical and alternate pathway. So these are all the structures of MSC class 1 and MSC class 2. So here we can see there is a peptide binding portion is present, and there is alpha and beta components, alpha 1, alpha 2, alpha 3, and beta 2 globulin is present in MSC class 1. And for MSC class 2, we are getting alpha 1, alpha 2, and beta 1, beta 2. Instead of alpha 3, in the MSC class 2, we are getting a beta 1 and beta 2. Coming to the acquired immune system, the development of acquired immune system is by fetal and neonatal life, that is some lymphocyte precursors from the bone marrow which enters into the thymus gland and become transformed into T lymphocytes. So some precursors also enter into the liver and spleen and get transformed into B lymphocyte. And that type of immunity, there are two types, that is cell-mediated immunity and humoral immunity. So the cell-mediated immunity is mainly by the T lymphocytes. Cell-mediated immunity happens by the T lymphocytes, that is by the help of the hormone thymosin, which is secreted by the thymus, which accelerates the proliferation and activation of the lymphocytes in thymus. So these thymus cells or T lymphocytes are divided into helper T cells or inducer T cell, cytotoxic T cell or killer T cell, suppressor T cell, memory T cell. And after transformation, T lymphocytes migrates and stay in lymphoid tissues present in lymph nodes, spleen, bone marrow, and gastrointestinal tract. And these T lymphocytes develop when an antigen from the invading microorganism is exposed to the T lymphocytes. The exposure or presentation of the antigen to the lymphocyte is done by macrophages, B lymphocytes, and dendritic cells. So, in antigen-presenting cell polypeptide products of antigen coupled to our major histocompatibility complex protein. And the antigenic products which activates the helper T cells and activated T cells in turn bring about the activation of the B lymphocytes. So activated helper T cells causes activation and proliferation of the cytotoxic lymphocytes and suppressor T cells, and also causes macrophages which also secreted interleukin 1, which promotes the immunogenic activity. So how the T lymphocytes or the cellular immunity functions, which the cellular immunity which protect against or which give protection against fungi and most of the bacterial pathogens, which produce allograft rejection in transplantation, and which causes delayed hypersensitivity, which is associated with autoimmune diseases, and also which provide tumor immunity. So the stages of cellular immunity, how these T lymphocytes are acting against the pathogenic agent, and which are all the stages are included. The first stage, as we said earlier, the first stage is the antigen processing and presentation. So the antigen which got phagocytosed and degraded by the antigen presenting cell or APC cells and are presented to the MHC complex. And this antigen is recognized by the lymphocytes. It is recognized by the T lymphocyte receptors. That is, the T lymphocytes are differentiated into CD4 and CD8 cells. So in this diagram, we can see that is in the infected cell is there is our antigen fragments are there, and the antigen is associates with the MHC molecule. Here is the class 1 MHC molecule, and it is attached to the T cell receptor. And the T cell receptor recognize the combination. And when the microbe enters, the antigen presenting cell, which it is present in the helper T cell. So there are two types of cells, cytotoxic cells and helper T cell. So in the T lymphocyte differentiation, the CD8 cells combined with foreign antigen and MHC complex activated and differentiated. Once it is combined with the foreign antigen, it is differentiated into cytotoxic T cell and suppressor T cell. So, when the CD4 cell combined with the foreign antigen and the MHC2 complex, which differentiated to helper T cell and delayed type of hypersensitivity. So, once the T cell is differentiated, let us see from first there is an antigen processing and presentation. Then the antigen is recognized by the lymphocytes and it is differentiated to CD4 and CD8 cells. From CD4 and CD8 cells, we are getting cytotoxic T cell, suppressor T cell, helper T cell, and also which produces delayed type of hypersensitivity. And after that, there is a T corporation will happen, which releases the differentiated T cell to release and also causes production of the T lymphocyte memory cells. After this phase, we are getting the attack phase. That is, the cytotoxic cells and natural killer cells produces perforin mediated killing of the pathogenic agent, and which causes lysis through the cytotoxic substances and causes apoptosis. So here the infected cell to the cytotoxic T cell. Here these are all the mechanism which is happening in the T cell, that is T to T cooperation is happening, which produces the perforins to release, and that causes the apoptosis. So here we can see the macrophage activation and causes inflammation, that is by the CD4 cells. So let us see how the cells are differentiated and what are the function of each differentiated cell here. So helper T cell which promote various activities of immune system, and it stimulates other T cells also and other B cells. And there are two types of helper T cells are present. They are Th1 cells which secrete interleukin-2 and gamma interferon concerned with cellular immunity. Th2 cells which secrete interleukin-4, interleukin-5 which are concerned with humoral immunity. So Th1 is for cellular and Th2 for humoral immunity. Cytotoxic T cell. The activated T cells comes into circulation to destroy the invading organism. How the mechanism of action, how it is acting. The membrane of cytotoxic T cells contain receptor protein to which the antigens or organisms tightly bind with. The organism is destroyed due to release of cytotoxic substances from the T cells. And the cytotoxic T cells can also destroy cancer cells, transplanted cells, tissues affected by foreign bodies. Next, the suppressor T cells, which suppresses the activities of cytotoxic T cells and prevents it from destroying the body's own tissue, which also suppresses helper T cells to further activation of the immune system. The memory T cell, some of the activated T cells do not enter the circulation but migrate to various lymphoid tissues and get activated when body is exposed to the same antigen again, like it is memorizing that particular antigen. So which produce the second time that produce the response is immediate and powerful. And specificity of the T cell means each T cell gets activated by only one type of antigen. So the surveillance system is happening in the body. That is, the body compartments are screened by the circulating WBCs. Here we can see a diagram. When the WBCs are contact with a cells, there are self markers are there. Our own body self markers are present, so there won't be any reaction and which is produced from the WBCs. But if it is come in contact with a foreign cell, there are markers are present, and that will cause destruction because it is recognized and causes our immune system to activate and produces destruction. So the lymphoid cells, the lymphocytes, that is around 25 to 35% of the WBCs. T cells are 70% of lymphocytes are T cells which produce cell-mediated immunity, and 20 to 25% of lymphocytes are B cells which produce humoral immunity. So coming to the humoral immunity, we have seen cellular immunity. Now let's see about the humoral immunity. B lymphocytes discovered in the bursa of fabricus in bird. At first, these B lymphocytes are discovered in the bursa of fabricus in birds. It is a major defensive mechanism against bacterial infection and viruses infected through respiratory and intestinal tract. So, upon entry of antigen, the B lymphocytes can bind with the antigen directly. So, they are transformed into two ways. They are of plasma cells and memory cells. The plasma cells form and secrete proteins known as immunoglobulins. There are IgG for around 70%, IgA of 22%, IgEm 7%, and rest is by the IgD and IgE. And memory cells which circulate through the body and remain dormant till the next exposure. So this coming to the sequence of events which causes the activation of B cells. The first stage, B cell recognize the antigen displayed on this surface of the antigen presenting cell with the help of its own surface receptor protein known as B cell receptor. After recognition, the B cell initiate a complex interaction between the B cell receptor and the antigen, and which activates the B cells. Same time, macrophages, that is the antigen presenting cells, which releases interleukin-1, which facilitates the activation and proliferation of the B cells. Activated B cells proliferate, and proliferated cells carry out the further actions. Simultaneously, the antigen bound to class II MHC molecules activates the helper T cells, also resulting in development of cell-mediated immunity. In humoral immunity, we should know about the antibodies. These antibodies are the proteins which recognize and bind to the particular antigen with very high specificity, which made in response to exposure to the antigen. So, one virus or microbe may have several antigenic determinant sites to which different antibodies may bind. Each antibody has at least two identical sites that bind antigen. They are known as antigen binding sites. Valence of an antibody. Number of antigen binding sites, and that is known as the valence of an antibody. Most of the antibodies are bivalent, and which belong to a group of serum proteins known as immunoglobulins. So this is the general structure of an immunoglobulin, which shows there are two antigen binding sites, and there is a heavy chain and a light chain. In the heavy chain, we are having variable region. And the light chain also, there is a small portion known as variable region and of light chain, but there is most part is a constant region. There is a one variable region and a constant region, and it is bound together by the disulfide bridges. See here, this is the B cell in a B cell receptor. Here we can see the heavy chains and light chains in the cytoplasm of B cell and in the cytoplasm of the T cell. The T cell receptor doesn't have a variable region or it is not a complex structure, and the B cell, which is having a Y-shaped structure. In the T cell, we are having a alpha chain, beta chain, and it is a single structure. Here, it is a Y-shaped double-stranded structure. So, here is in the antibody B, there is a antigen binding site, and antigen binding sites are properly binds to the antigen, and they are the antigenic determinants known as the epitopes. So the antibody structure, let's see, it is monomer, that is a flexible Y-shaped molecule with 4 protein chains. There are 2 identical light chains and 2 identical heavy chains. There is a variable region and a constant region. The variable region means 2 sections at the end of Y's arm contain the antigen binding site and identical on the same antibody but vary from one antibody to another. The constant regions are the stem of monomer and lower part of the Y arms. FC region, stem of monomer only. That is important because they can bind to a complement or cell. So let's see the structure of each antibody. That is, first one is IgG. It is a monomer, which is the most abundant immunoglobulin class in blood, also present in tissue fluids, which promote opsonization, neutralization, and cross-linking of the antigen, which is less effective in activation of complement system than IgM. Remember, which is the only immunoglobulin class that crosses placenta, thus conferring passive immunity on fetus. So, structure is a monomer. It is of 80%. Location, it is present in blood, lymph, and intestine. Half-life in serum, it is around 23 days. And they process complement fixation and also which shows placental transfer. It enhances phagocytosis, neutralizes toxins and viruses, protects fetus and newborns. Coming to the immunoglobulin M, that is a pentamer that is having 5 units and it is connected by J chain. It is produced after initial exposure to the antigen. Then its concentration in the blood declines, which promotes neutralization and cross-linking of antigen, very effective in complement system activation. So, in a nutshell, it is a pentamer. It is around 5 to 10% in serum. Look, it is present in blood, lymph, B cell surface. Also, it is a half-life of 5 days, which is process complement fixation, but there is no placental transfer. It is the first antibody produced during an infection. It is the first antibody and which is effective against microbes and agglutinating antigens. Next, coming to the immunoglobulin A, which is a dimer. And it is having J chain, which combines both the parts and also which is having a secretory component. So it is present in secretions such as tears, saliva, mucus, and breast milk, which provide localized defense of mucous membrane by cross-linking and neutralization of antigens. Presence in breast milk confers passive immunity on nursing infants. So, immunoglobulin A is a dimer which is present in serum around 10 to 15%, which is present in secretions and blood and lymph. Its half-life is 6 days. There is no complement fixation and which is not transferred through placenta. And it produces localized protection of the mucosal surfaces which provide immunity to infant digestive tract. The next one is immunoglobulin D, which is a monomer. It is present primarily on surface of the B cells that have not been exposed to antigens. So it acts as antigen receptor in the antigen stimulated proliferation and differentiation of the B cell, that is known as clonal selection. So monomer, which is present around 0.2% in the serum, which is present in the B cell surface, blood and lymph. Its half-life is 3 days. This also produces no complement fixation and no placental transfer. So in serum function its a serum function is unknown, but on B cell surface which initiate immune response. And the last one is immunoglobulin E. This also a monomer which is present in blood at low concentrations. It triggers release from mast cells and basophils of histamine and other chemicals that cause allergic reactions. So, coming to the immunoglobulin E, it is a monomer. It is present is very minute quantity, that is 0.002 percentage. And it is bound to mast cells and basophils throughout the body in blood. Half-life is 2 days, no complement fixation and no placental transfer, which is more prominent in allergic reaction, also which is present or which is get activated during the lysis of worms. And the coming to memory B cell. The memory B cell remain inactive until the body is exposed to same antigen for the second time. Just like our memory T cells, the antibodies produced on first exposure is in less concentration, which takes longer time for production and belong to mainly of IgM type. But on the second exposure, the antibody production is faster, more in concentration, and belong mainly IgG type. So first IgM, second time it is IgG, and this forms the basic principle of vaccination against infection. So this is the graph which shows the primary and secondary immune response. Here we can see the IgM. It is a very first immunization. It is a very short period and it is very low concentration. And in the second immunization or in the second exposure, there is also IgM will produce, but the concentration of IgG will be very high, and it is a sharp reaction. So during the second time, the immune system will regain very fast. The plasma cells which secrete large quantities of antibodies, that is around 22,000 molecules per second, and these antibodies are produced till the end of lifespan of plasma cells. That means around several days to weeks. And antibodies are gamma globulin fractions of the plasma protein that forms around 20% of the plasma proteins. So coming to the apoptosis, we have seen that the cells may produce apoptosis. Apoptosis means that it is a programmed cell death or a falling away. The human body makes around 100 million lymphocytes every day. So if an equivalent number doesn't die, that will cause leukemia. So the B cells that do not encounter stimulating antigen will self-destruct and send signals to phagocytes to dispose of their remains. And many viruses infected cell will undergo apoptosis to help prevent spread of the infection. So once the antigen bind to antibody, there is some complex reactions will form. That is affinity. That is, once the antigen bind to antibody, there should be some affinity to the antigen and antibody, and which leads to agglutination Antibodies cause antigens to clump together. That is, IgM is more effective than IgG, and that is heme agglutination. Agglutination of the red blood cell used to determine ABO blood group type to detect the and also which detect influenza and measles viruses. Next stage is opsonization. Antigen is covered with antibodies that enhance its ingestion and lysis by the phagocytic cells. So, we see that there is agglutination, opsonization, neutralization, and which causes activation of the complements, leads to inflammation and antigen-dependent cell-mediated cytotoxicity. So, the third stage is neutralization. Neutralization means immunoglobulin inactivates viruses by binding to their surface and neutralize toxins by blocking their active sites. And the fourth stage is antibody-dependent cell-mediated cytotoxicity. So that used to destroy large organisms like worms. That target organism is coated with antibodies and bombarded with chemicals from non-specific immune cells. And finally, which produce complement activation, that is IgG and IgM trigger the complement system, which result in cell lysis and inflammation. So let's see it in detail. First, the stages are antigen processing and presentation, recognition by the lymphocyte, lymphocyte activation, production of antibodies, and inactivation of antigen or attack phase. There are two phases, that is direct and also through the complement system. First stage is antigen processing and presentation. Here we can see a diagram. So antigen in the first exposure engulfed by the antigen presenting cell, which stimulates cytotoxic T cells, helper T cells. Also, the antigen will directly stimulate the B cells. Coming to the B cell differentiate into plasma cells and memory B cells. In the same time, the T cells also will get activated, and which forms the memory T cells and helper T cells, and also the cytotoxic T cells differentiate to form memory T cells, also which activate the cytotoxic T cells. And the second, that is coming to the humoral immunity, that is antigen presenting, and it is differentiated into B cells and T cells. Second, by the recognition by the lymphocytes, the antigen presenting cell, that is the class 2 MHC molecule, which is engulf the foreign organism or the bacterium. In the B cell, the immunoglobulins will receive the bacteria and which get activated. So which produces clone of plasma cells and clone of memory B cells, which secrete antibody molecules. The lymphocyte activation and production of antibodies. The clonal selection of B cells is caused by the antigenic stimulation. So the antigenic receptor, there is produces a variety of B cells are there, and there is antigen receptors are there, and these cells will proliferate. and which proliferate clone of plasma cells and clone of memory cells, which also causes antibodies secreted into the circulation. And how the antibodies are produced? The B cell, which develop from stem cells in the bone marrow of adult. After maturation, B cells migrate to lymphoid organs. The clonal selection means when a B cell encounters an antigen it recognizes, it stimulated and divided into many clones known as plasma cells, which actively secretes antibodies. We have seen the diagram earlier. And each B cell produces antibodies that will recognize only one antigenic determinant. And clonal selection means B cells or T cells that encounter stimulating antigen will proliferate into large group of cells. So we have a question like, why don't we produce antibodies against our own antigens? Because we have developed tolerance against to that particular our own cells, and also which produce clonal deletion. B and T cells that react against self-antigen appear to be destroyed during fetal development. The clonal deletion process is not fully understood till today. And there is immunological memory is present, that is antibody titer, that is the amount of antibody in the serum, and the pattern of antibody levels during infection, that is the primary response. That is, after initial exposure to antigen, no antibodies are found in serum for several days. A gradual increase in titer, that is first of IgM, then of IgG is observed, and most B cells become plasma cells, but some B cells become long-living memory cells, and also which produce a decline of the antibodies. And during the secondary response or in the second response, the subsequent response to the same antigen displays a faster and more intense antibody response. The increased antibody response is due to the existence of memory cells, which rapidly produce plasma cells upon antigen stimulation. So coming to the stages, in the final stage is inactivation of antigen or attack phase, that is direct or through the complement system. So the mechanism of action, that is direct action, we have seen like agglutination, precipitation, neutralization and lysis. And through complement system, there are two ways, that is classical pathway and alternate pathway. In the classical pathway, again, there are few steps are present, that is opsonization, lysis, chemotaxis, agglutination, neutralization, and mast cell activation. In alternate pathway, there is the binding of factor 1 with the polysaccharide and activation of the enzyme C3 and C4 will be produced, and that will causes the destruction of the antigen. So here we can see antigen of first exposure which stimulate the B cell and which is engulfed by the antigen presenting cell, which also stimulate the helper T cell, which produce activation of the memory T cells and helper T cells, Also, the B lymphocytes will differentiate or give rise to plasma cells and memory B cells. All these together which secretes antibodies and defend against extracellular pathogens. So how the direct action is produced? Antibodies can inactivate the invading organism, that is the bivalent nature of the antibodies and multiple antigen sites on the invading organism. And it will happen in one of the following ways, like or agglutination, precipitation, neutralization, and lysis. So, binding antibodies to antigens inactivates antigen by neutralization, that is neutralizing, as we see earlier, blocks viral binding sites, coats bacteria, or cause opsonization. And agglutination of antigen bearing particles like microbes, precipitation of the soluble antigen, or also produce complement fixation, that is the activation of a complement, which all these processes enhance phagocytosis, like neutralization, agglutination, and precipitation enhances phagocytosis by the macrophages, and the complement fixation or complement system activation will produce or which leads to cell lysis. The complement system is a collective term to describe a system of about 30 different proteins. They are the enzyme precursors, and this is a stronger action than direct action. That because the complement system accelerates the fight against invading organism. The principal actors in the complement system is the plasma enzymes. They are C1 to C9, B and D, and these enzymes are in inactive form and get activated by two major pathways. They are classical pathway by immune complexes and alternate pathway which is stimulated by bacteria, virus, or fungi. So, pathway of the complement system C1 to C9 is activated in a pathway. Once C1 is converted to C1, activated C1 by the antigen antibody binding complex, and it leads to a long system of activation, and finally it activates the C9, which causes the lysis of the cells. The classical pathway, which activated by antigen antibody reactions. That means once C1 binds with antigen antibody complex and triggers a series of events to activate other enzymes, that is, which produce multiple end products, and that are also having important effects. So the complement pathway also produce opsonization by C3b, lysis by C5b, 6, 7, 8, 9, chemotaxis C5a, agglutination, neutralization, activation of basophils and mast cells by C3a, C4a, and C5a. Coming to the alternate pathway, activation of alternate pathway which triggers due to blinding of properdin with the large polysaccharides present in the cell membrane of the invading organism. This activates C3 and C5, which ultimately attack the organism. So the humoral mediated immunity, which is having plasma cells and cytokines, which produces the T cells. Let's see the changes. B cell, which is presented, T cell, so coming to the organ transplants and rejections, there are mainly 4 types of organ transplants are present. They are autograft, isograft, allograft, and xenograft. So autograft means tissue graft from one body side to another in the same person. Isograft, which is the graft received from a genetically identical donor, that is identical twin. Allograft, that is a graft received from genetically non-identical donor of same species. Xenograft means graft received from another species of animal. So the transplant rejections are mediated by the immune system, especially T, C, and natural killer cells, and also by the antibodies. Autograft and isograft is corrected by the MHC compatible. Xenograft, it is most MHC compatible. Allograft, attempt to obtain the best MHC match to avoid the MHC or transplant rejection. The immunosuppressive therapy, which used to delay or prevent the rejection, like corticosteroids, which suppress inflammation, anti-proliferative substances, which prevent or kill rapidly dividing cells, immunosuppressants, which prevent or kill rapidly dividing cells, and the side effects of these type of mechanism will be harsh, and which causes increased risk of infections. Coming to the immunological dysfunction, there are 2 types of immunological dysfunctions we can see, like congenital or genetic, and that is of varied inborn errors. Also, there is a type like acquired by drugs, also produced by cancer or malignancy, and which can be viewed as a failure to immune surveillance, Hodgkin disease, and also by AIDS or HIV, which kills the Th4 or T helper cells. So autoimmune diseases. Autoimmune disease, that is the production of antibody and T helper cell against self tissues. They are the examples and tissues affected are multiple sclerosis. They are white matter of the nervous system got affected. In Graves' disease, thyroid cells will get affected. Type 1 diabetic mellitus, beta cells of pancreas will get affected. Systemic lupus erythematosus, anti-DNA kidneys, heart and lungs and skin are getting affected. Rheumatoid arthritis, which destroys joints and cartilages. Glomerulonephritis, they produce impaired renal function, may be secondary to other autoimmune diseases. So mechanism of immunological dysfunction, that means the failure of lymphocyte programming. New self-antigens are produced because of gene mutation or structural change of the antigen, produce haptens or by infection. Foreign antigen that closely resembles self-antigen, resulting in cross-reactivity. And the hypersensitives. Hypersensitivities or allergies, that means the immune system responds to a harmless substance as if it were a threat. That is, allergen, that is the antigen of an allergic response. Allergen is the substance which is known as the antigen of an allergic response. That is mainly happens by the IgG antibodies. So the IgG antibodies attach to mast cells in the body tissue and also by the circulating basophils, and that will produce the allergic response. So on the second exposure, what will happen is more of the same antigen inverts or antigen or allergen invert the body. The antigen combined with IgE, which is attached to the mast cell, which triggers the degranulation and release of histamine. And with the histamine cause blood vessel to dilate and become leaky, and which promote edema, stimulates the secretion of the large amount of mucus, which causes smooth muscles to contract, especially in the respiratory system, and also which cause outpouring of fluid from the capillaries. So sometimes it may cause itching or swelling and release of mucus from the respiratory system. Constriction of the small respiratory passages will happen. So immediate hypersensitivity is type 1. The symptoms within seconds of exposure to an allergen, that is requires sensitization, means there is a previous exposure. So that is known as anaphylaxis, IgE mediated, that is by mast or basophil cells. Local irritation means by histamine induced vasodilatation and increased permeability, leads to watery eyes, runny nose, itching and redness in respiratory system, which produce allergy-induced asthma. In systemic anaphylaxis, there is anaphylactic shock, which associated with allergen that have systemic distribution, widespread vasodilatation and airway swelling. So the term atopy means the tendency to display type 1 symptoms to certain environmental antigens without prior sensitization. The hypersensitivity type 2 and 3 means subacute hypersensitivity, that is by the IgG and IgM mediated. Like cytotoxic reactions, type 2. Antibodies bind to cellular antigens promoting complement fixation or inflammation or phagocytosis. Or immune complex H or type 3, which is widely distributed antigen, reacts with antibody. And this antigen antibody complexes cannot be cleared, which produce persistent inflammation or tissue damage, like farmer's lung associated with autoimmune disorder. And type 4 hypersensitivity, that is known as delayed hypersensitivity or cell-mediated hypersensitivity, which takes 1 to 3 days to react. Which involve
cytotoxic T cell, helper T cell, Th1 cells and macrophages, allergic contact dermatitis, that is by the poison ivy, heavy metals or a TB tinate test, and agents which act as haptens and elicit response after binding to tissues. Next, we can see about the acquired immune deficiency syndrome or AIDS, which is caused by the human immunodeficiency virus discovered in 1983, which specifically targets and kills T cells. The normal body cells are not affected, but the immune response is not launched. The HIV virus doesn't kill you because it cripples your immune system, and the immune system shut down common diseases that our immune system normally could defeat become life-threatening. Can show no effects for several months and all the way up to 10 years. So in a nutshell, let's see the schematic diagram which shows the T lymphocyte and B lymphocytes in immunity. The bone marrow lymphocyte precursors are differentiated into T lymphocytes and B lymphocytes. T lymphocytes are present in thymus and B lymphocytes bursa equivalents like liver and bone marrow. The T lymphocytes releases helper T cell or CD4 cells, which activates the B lymphocytes and forms a memory B cells. Also, the helper T cell activates the T lymphocytes, which differentiates into cytotoxic T cells, mostly CD8 cells and memory T cells. Also, which produces suppressor T cells. In the T cells, these CD8 cells will produce cellular immunity, and the plasma cells differentiates to form immunoglobulins. Immunoglobulins, group of immunoglobulins, and that will lead to humoral immunity. So this is a nutshell of our immunity and immune system. So let's see a small differentiation of the immunity, cellular and humoral immunity. Cellular is T lymphocytes. It is processed in thymus. Exposure to antigen produces helper, suppressor, memory, and cytotoxic cells. Lymphocytes burn holes in the cells to be destroyed by the releasing substances like interleukin, prostaglandins, etc. Humoral immunity by B lymphocytes, which are processed in liver and spleen. Exposure to antigen produces plasma and memory cells. Plasma cells synthesize and release free antibodies into the circulation. 80% of circulating lymphocytes belongs to the cellular immune group, and which is having role in responses against bacteria, fungi, viri, and against transplanted organs and tissues. Humoral immunity is around 20% circulating lymphocytes, which belong to only 20% and have role against bacteria only. So by this, we finish our immunity. Thank you.